Welcome to the Cell-to-Cell Communication in Development module. In this talk, we're going to learn about juxtacrine signaling.
So far, we have looked at paracrine signaling, but there is another type of signaling that occurs during development, which is juxtacrine signaling. Juxtacrine factors are not secreted and usually remain on the cell membrane of the source cell and interact with cell surface receptors and target cells. We're going to look at three different examples of juxtacrine factors. The first one being the notch proteins that can bind to the ligands that are called delta, jagged, and serrate. Another example of juxtacrine factors are the adhesion molecules like cadherins and the EPH receptors and F-brand ligands. Out of all of these, we're going to focus on the notch pathway. Now, notch is a cell surface receptor that can bind to the ligands delta, serrate, or jagged. As shown in the figure here, we can see notch is actually a cell surface protein, and it is associated with the protease that is present on the intracellular part of notch. This is the receiving cell that is going to be getting the actual signals. Now, the signaling cell, which is the one that is actually making the ligand, which in this case is delta. Now, delta, as you can see, is also a membrane protein and hence it is not secreted. When delta is present in the signaling cell, it will interact with notch. The interaction of notch with delta causes a conformational change in notch, and now, upon ligand binding, the cytoplasmic domain of notch can be cleaved through the action of that protease that was associated with the cell membrane. Once the notch protein is actually cleaved, the cleaved fragment enters the nucleus to stimulate transcription. Notch is known to be an important factor for the development of the nervous system and also plays roles in immune cell development.
Thus, to summarize, we learned about the notch pathway and its role in juxtacrine signaling, as well as what is lateral inhibition.